Okay, we're going to go ahead and get started. Hi, and welcome to Machine Learning for Medical Devices. My name is Nicole Owens, and I'm the Director of Marketing Communications for MassMedic. If you're not familiar with MassMedic, we're a medical device trade association for companies in New England, and we work to bolster the ecosystem through educational events like this, connection, advocacy, and awareness. Today, I'm really pleased to bring you this webinar from our member and friend, Sunrise Labs. And we have two presenters today. I'll get to them in just 1 second. I want to let you know that we will be taking Q&A throughout the presentation, and we'll have some dedicated time at the end. So please feel free to utilize the chat and the Q&A function. So with that, I am happy to introduce Adam Jacobs. He's the Chief Technology Officer, and Bob Boudelier, the Director of Sunrise Labs, Southern New England Office and Technical PM. And with that, I will turn it over to Adam and Bob.
Thank you. Welcome, everybody. I hear we have a good turnout, so that's exciting. Obviously, machine learning is a big field these days, and it's getting more and more utilized in medical devices. Personally, I've been involved in building automated classifier systems much of my career, along with building other medical devices. So we felt that we had some knowledge to share, and it was time to share it. Bob, would you like to introduce yourself?
Sure, I'd like to. So thank you, Adam. So Adam has far more experience in machine learning than I do. But I have been involved in 5 different projects, programs that have had machine learning in them. And I pretty much take every course I can get my hands on to learn more about machine learning. So it's a continually growing field. And while there are far more examples, relevant examples in the field outside of medicine, it is now finding its way into great applications in healthcare as well. So I look forward to sharing what we've learned along the way, and not all positive, some of it negative, but that's how you learn. So without further ado, I think we can get started.
Okay, great. A brief introduction to Sunrise Labs. We are a medical device development company. We do the engineering and development of a wide range of medical devices. And we're about 110 people, and we're in Southern New Hampshire, not close to the Boston area, which is a good area to be for medical devices. It's sort of a full service group. We tend to do more complicated things, electronics, software, mechanics, optics, full system development. So anybody who's interested can look us up. And let's go to the next slides. Just to introduce the topic, as machine learning gets used for more and more things, it seems, at least in our opinion, that there are some times when it really helps, and it can be very, powerful. And there's also people who try to apply it for everything, and there's tricks that we've learned about how to apply it, but also when to apply it. So that's the general topic here, and we'll try to sprinkle in some actual experiences to point out the points that we're making. So just a brief note on terminology. So when you think about artificial intelligence, that's kind of the broader field. Often artificial intelligence, at least historically speaking, first of all, not everybody agrees on what it is. So I guess this is my version, along with some other people's. One of the principles of artificial intelligence is that that it's not a fixed thing, that there will be automatic learning as new information is put in. So and those are dynamical systems where you can continue to upgrade an algorithm. That's one of the properties that an artificial intelligence system may have. And another one is that it just feels like human intelligence because of its ability to do that. You know, you've heard the Turing test. It sounds like a person. If it sounds like a person, then you've met the Turing test, and you can fool somebody with the responses. So that's not really the focus of what we're going to talk about now. Most of the work that at least we've done has been on building classifiers. Classifiers is when a machine is able to take inputs and decide on what the class of the or some kind of a number, what kind of a score of the inputs is. So, for example, if something's cancer or not cancer, and I personally worked on a project called Melfind for over a dozen years, and it was a multispectral imaging device. So our goal was to say whether or not we thought it was worthwhile to do a further biopsy. So that was a go no go kind of decision. Search engines, machine learning search engines, are ways to search through large spaces of data and possibilities of how to process that data to try to come up with either classes of classifiers or regressors that will come up with a score. And it's one of the tools that one uses to create these applications. One of the so they'll try many combinations. You know, you've probably heard of this part. And I apologize for the early terminology because many of you probably know some of this. One thing to note is the all important thing of the merit or loss function, which is used to quantify how well you're doing, and that's how it chooses which of the solutions in a search is better. So just some basic terminology. Go ahead, Bob. So again, the basic stuff here. Basically, you have data, and you train your classifier on that. You split your data into two sets: a training set and a test set. You then train your classifier repetitively over and over again until you get the maximum merit function, and then that's basically what are called your training results. At which point you would go to the next slide and then you test the results on the test data, and you see how well you did, and they're often not the same. So that's, and there's lots of details here that I'm glossing over. People can split up data sets in multiple ways. And we'll talk a little bit about that too, because there's pitfalls there, and that's the basic idea. And again, just as basic for the people who don't know all the basics. Next slide. So in our experience, there's different kinds of learning. There's really three kinds, but the major two that we tend to use is supervised and unsupervised. Supervised is usually what we've done, and that's because there's a certain result that you want to get. I apologize for the background noise. There's actually construction going on, and I'm in a hotel, so I apologize. So supervised learning is when you can give something an answer, and then it'll know what the ground truth is so that it can train to that. Otherwise, it doesn't really know what's what. If you want to say something's cancer versus not cancer, the machine learning algorithm has to know while it's training. So most of it is supervised learning in medical devices. Although there are applications that are not. Sometimes, especially the unsupervised learning often tends to be with large databases, where you're looking through data, trying to see what's interesting in a way. What patterns there might be, that what correlations there might be. Say you have hospital records, and you start to look for combinations of things that that are interesting. Devices typically are looking for something specific, and that's why typically you'll use supervised learning.
One thing I'd like to add to that, the FDA considers supervised learning locked type of learning, so that you can validate the output and it doesn't change over time. Where if you have reinforcement learning built into the algorithm as well, then they consider that adaptive. So they have different criteria that they will apply to review of your product. They would like to see us apply different criteria when we submit a product for review, to make sure that we tell them if there are adaptive components, then they want you to treat that with a different level of care to make sure that the adaptations are, the outputs of that are tested, in a rigorous way to make sure that there are not surprises in the field.
Right. Most of the submissions that we've done or I've done have been when you come up with a classifier, you test it with test data that gives you a real result that's statistically significant, so that the FDA can know what to expect. And that's why typically in the past they've liked things to be locked down. It is sort of the simpler path, though they've been looking at more dynamic classifiers and ways to deal with that, and most of that's by dealing with a process. And we'll talk about that a little bit later too. It brings up the idea of a gold standard. The gold standard is the ground truth for data that you're going to use during supervised learning. And that might be, for example, an existing instrument, like let's say it's a lab test, and it's the best lab test people know about. So that you would compare your device to that and to see how it does. Or sometimes there'll be a panel of experts, for example, pathologists. What we did is we had, in one, we had 5 pathologists and they voted. And the reason they did that is because they didn't all agree. We learned a lot about pathologists during that study. But that's a gold standard that you would use. Often standards are not that gold. They often have weaknesses in them, and a big part of the learning is to come up with a decent gold standard. And if you're doing something new, then it's even harder, because it may not have a real standard at all. It brings up a point here, which is that more data isn't always helpful, because the data, for example, this is something that we've seen a lot, is that people talk about having a connected device, for example, which is another thing that's happening a lot. And so you're collecting data during this whole time. And sometimes that's useful because you're getting more data, but that's only really useful in a supervised learning situation when you have a gold standard to classify it by. So it's not always as helpful as people think it's going to be. Just it's a thing to watch out for. You go and you do this study, and it's a double-blinded study. You end up generating a classifier, and you present it to the FDA, and now you're online and you're getting tons of data from everybody who's using it, but there's it's not in a controlled environment, and there's not necessarily a gold standard. So the gold standard is a really key part of this whole thing that is often overlooked. Next slide. So what are some issues with doing machine learning for medical devices? The most obvious one is that there's small data sets in the beginning. If you create a device and you put it out there, it's almost always harder to get data than one thinks it's going to be. And so people can put out, in a normal medical device, if you look at Gaussian distributions, you might need a few hundred patients, or maybe high hundreds of patients. And for machine learning, those are small numbers typically. So so you'll get early results with small data sets, and they essentially can be misleading. So getting the kind of quantities that one needs can be difficult, which leads to the next issue, which is overtraining. Or overtraining is when you you give the classifier enough degrees of freedom to essentially draw the boundaries around the data. So I have two analogies here. One is gerrymandering. With good gerrymandering, you can make any election outcome you want. And the same is true with, oh boy, I suppose you can hear that. With gerrymandering, you can make any result you want, and that's what the classifiers do. They will gerrymander their solution so that you get the best results. The other analogy I have is drawing the bullseyes around the darts. So you throw the darts against the wall, and if you have enough bullseyes, you can get 100% accuracy because you have as many bullseyes as you have darts. But that doesn't tend to generalize when you start to get more data. So that's a big problem that everybody knows about who's in machine learning, and there are some ways to deal with that. I really apologize for this.
That's all right.
It sounds terrible here.
It's not that. It's actually not that bad over here, and we do have some questions too when we get to a point.
Okay, great. So after this slide, sometimes the available data is not applicable. People will use data that there's differences between the actual use cases or the machine and the device. And finally, repeatability. People really underestimate in a device how important repeatability is, and I'm going to talk about that a little more later. If you have multiple instruments, you might go out with 5 instruments to gather data, and then you make 1,000 instruments, and those 1,000 instruments likely will not be in the same range that the 5 instruments you had in. So there's also some things that are not repeatable, and sometimes classifiers are built requiring some kind of repeatability. Like, for example, if you put like temperature is a big one. Sometimes temperature is not controlled. Sometimes, for example, if you take a picture of something, that picture can rotate, and if your classifier is sensitive to image rotation, that can be a problem. So there's things that you have to be careful about with repeatability. Next slide.
There we go.
So another issue is finding the right application, which is what we're going to talk about. What are the properties of kind of applications that seem like they're useful? And I will not say that it's always easy to tell, but there are some things to look out for. Outliers are a big thing with small data sets. You will get rare-ish events that are problematic. So outliers especially with smaller data sets, is a problem. Bob, you have the next one's for you, really.
Yeah, one of the things that we've run into a number of times is an application comes up. There was one with involving a toothbrush, which is oddly enough a medical product. Anything that you stick in your mouth these days, including a tongue depressor, has an FDA class associated with it. And the objective was to use a support vector machine to determine what part of the mouth you were in as you were brushing your teeth. And the latency of the processing was too long. You were in a different part of your mouth by the time you got the answer of where you used to be when the picture was taken. So it's important to structure when you do the architecture, make sure that if latency is going to be a problem, that either the processing is fast enough to be able to give you the answer in time, or use other means to tell where you are. And that was in fact the outcome of the original testing. Latency was too long to give you an accurate determination of where you were by looking at the teeth that it saw, to be able to update an app in real time and show you in fact where you were using that method. Oh, you're on mute.
If you read some of the literature on predictability, for example, there are certain classes of things that are easier to predict than others. And people keep pushing that boundary, but the classic example is the weather. The weather reports have gotten predictably, the predictability in terms of the number of days that they can predict out has grown over the last 30 years. And so there's properties of weather it's a physical system. We get more and more measurements. We get more, more sophisticated models, and that's gotten better and better. There are some things where there are that people have not gotten better on, and the one I like to quote is the stock market. It's a very complicated system. It has millions, if not billions, of inputs and people, and there's lots of inputs into it that can be random. For example, black swans that people have talked about. There'll be a war somewhere out of nowhere, and people don't expect that. So there are classes of problems that are more suitable, and hopefully we'll have a few thoughts on that later. Go ahead, Bob.
One of the things I wanted to bring up here, this pre-sorting of data for startup issues. The Mayo Clinic had discovered an apparent connection between ECG T waves and potassium levels. So patients with high potassium levels, there was a apparent change in the T wave, and they wound up partnering with AliveCore and giving AliveCore a whole bunch of data to run a machine learning algorithm on that. And the data was pre-sorted for outpatients. And when they ran all the analysis 2 months into it, they couldn't find the same correlation that the team at the Mayo Clinic had found. So they asked, could you give us the entire ECG for all these patients, and then both inpatients and outpatients data? When they ran that, they got a less than 1% error, and they had good correlation. So pre-sorting for just the T wave data and just outpatient data actually gave them a negative result, and they got a positive result by just including all of the data. So that was a learning that was presented in Dr. Eric Topol's book, Deep Medicine. So it's an excellent resource for a lot of current work that's going on in machine learning.
So we do have a question. I think you went too fast on that slide there, Bob. But we do have a question. It says, can you talk about the element of time validity of a measurement? And can you use intermittent data that is, for example, an hour old or a day old? I think it depends. I hate to say it depends on the application, but of course it does. There are some things that are just diagnostic. You take a blood test, and you're going to measure that blood test. Obviously, if you're doing measurements on patients in vivo, and for example, patient monitoring data, ECGs or multi-parameter data, that obviously does have a shelf life because the person is a dynamic health state. So I don't tend to see problems with aging for making a classification of the data. It's more a question of, does the application care if it's old data? And we have done a lot of patient monitoring kind of applications. I was particularly interested in multi-parameter monitoring because, and we'll talk about this later, about getting multiple inputs into a classifier. Because, for example, let's say you wanted to classify a cardiac event, maybe VFib, which is the big one, of course. And there's artifacts and things in the ECG that can cause false positives to happen and cause your specificity to go down. Getting more types of data to correlate to is one of the ways that we reduce false alarms, because our alarm fatigue is a big deal in a hospital, and nurses sit there all day long and they get false alarms. And even with high specificities, when you have low incidence of things, you can have very high false alarm percentages. So I would say that, to answer the question, it really depends on the application. In general, the classifier doesn't really care. So, all right, so getting back to the startup issues, we talked about overtraining. And there's a thing called k-fold cross-validation, which I'm sure many people know about. It basically takes your data set and cuts it up into different pieces and builds a classifier for the different slices through the data, and then compares them and tries to use to get a better answer for that. And sometimes that's the best you can do, because I've seen many applications where they have 70 or 30 or 40 or 100 data sets, and it's not anywhere near enough. So they do what they can do with that data, and then they use k-fold cross validation. I don't tend to trust it very much, because you are kind of mixing up your training and data sets. But as a first step, people do it because it's the best they can do. Gold standard or instrument design drift is a classic one. When you start out, it's hard to get the perfect instrument the first couple of times, and then the more you learn, you see what matters. You learn what parts of your instrument. Maybe it's temperature. Maybe it's I'll go back to the melanoma project, where we were taking multispectral images of melanomas, and it turned out that the focus of the lenses really mattered. So trying to get a lens that has perfect focus across a big field of view, across the wavelengths that we were using, which was from 400 to 1,000 nanometers, which is a very wide wavelength. It turned out we could do it, but it was difficult to make. So it took a number of years to get the repeatability of the instrument, plus the calibration techniques to calibrate out things that mattered. So instrument repeatability is an often overlooked way to things that will affect your classifier results, and we're going to talk about that in a moment more. The gold standard drift can happen as well. A classic example is somebody has what's called the golden unit. It's okay. We used the unit to build the classifier. Let's make all the units just like those units, which is kind of a recipe for disaster. One, because it's really hard to make everything exactly the same across instruments, and also instruments age. I mean, things like you could have a phantom. Let's say the phantom is an image, maybe even a photographic image. If it's that photograph can age, and over time it can get torn, or phantoms can they can age for various reasons. So it depends on the application. The same thing is true with so the same thing is true with certain biological compounds. The age of it, for example, might matter. And the final point is that a common issue is that prototype data that you take during early data collection is sometimes different than where you end up. You might, so you'll make a first prototype, you'll gather some data, you spend a lot of money going to somewhere and a lot of effort, and you gather some data. And of course, you want all the data you can get, because data is hard to get. We just talked about that. So, but then you go and you make the instrument different or better, and you've now enlarged the variability of your data. So, if you can, it's often a good idea to expect to throw away very early data and try not to save it. So next slide. So what are some properties of things that make for a good application? And we'll talk more about this later. One is, if you're lucky enough to have something where it's easy to obtain the data, sometimes data is easy to get. I mean, for an example of that is that there are publicly available databases. Some of you might know PhysioNet, which is an MIT computational physiology data set, and they, so that's an easily available data set. And there might be pathology slides. There are many groups that have stored pathology slides with diagnoses. So if you're going to scan some slides and try to build a classifier on it, you could go out tomorrow and get 100,000 slides with diagnoses. So the easy to obtain data is probably the most important thing, I think, in this field. what gold standards are. I've always been surprised how gold standards are more interesting than one would expect. So in cancer, for example, cancer diagnosis, there's multiple thoughts of what cancer is. There's multiple schools of thought where one example might be that it's a genetic disease of a cell, and you could sequence that cell and say, oh, that has a genetic disorder, that's cancer. And then another school of thought is that, well, actually, you're often getting genetic disorders in your body, and you have a feedback system to control that. And cancer is when there's a certain level of disorder in the organization of many cells. So you look at the pathology, and some pathologists will look at, what does the nucleus look like? And some pathologists would say, no, well, the body might kill that cell, and it most likely will. You're often getting those kinds of cells. So I look for certain patterns that show that the body has lost the ability to control, and that shows up in different, say, patterns of cell organization. So it's not always obvious what the gold standard is, because it's not always obvious what a disease is. But you're going to have to pick one and go with it. So the next one is that if there's an instrument or a technique that causes a lot of variability, you can accommodate that variability, but it tends to make the results less accurate, lower specificity and sensitivity. So if you can do something where the classifier really doesn't care about the technique, or things like in an instrument, maybe temperature really doesn't matter in this particular thing, or you can make say it's a chemistry test and temperature really matters. Well, then you have to do something where you're really controlling the temperature, and maybe it's hard to control the temperature. So making it not sensitive to the instrument or technique is really a good thing. And finally, knowing what is good enough. This is another problem, and there was a classifier that we built that I was involved with, and the FDA. If you look at the specificity of the current practice, it's about 2 to 3%. People take many more tests than are positive. And when we train the classifier, we said this was again a cancer diagnostic, and we don't want it to be, we don't want to miss anything. That's the worst sin. So we trained it for 100% sensitivity, which drove the specificity down, but it was still 3 or 4 times better than the state of the art, and it had a higher sensitivity. But the specificity isn't high. It's just better than the existing standard. And that still had problems. It was a big to do about it. So knowing what's good enough, you might think that, but the market might not agree. So be aware of that. So this next one is kind of a hard one. Do you have adequate information in the data for the classifier? And you don't always know that. I mean, part of when you're doing things like neural nets, especially, or you don't always really know what they're looking at. But let's say, for example, that you're expecting to have, let's say, expecting to diagnose a genetic disorder, but you're looking at, I don't know, a color, like a spectroscopic signature. Does that single genetic disorder show up in the spectroscopic signature? Well, maybe not. So people often try to use spectrography or images or some kind of chemical reaction or biological reaction, DNA. And the question is, do you have the information from this test, all the information you need really to know what that disease state is? So, and you can do sort of a top level thought a Gedanken experiment. Do I think I have it? If you're looking for a really specific genetic disorder, You might not if you're looking at the color of the tissue, for example. So, or maybe you do because you believe that it's a macroscopic property like we talked about before. So it's not always obvious, and it's not always, but it doesn't mean you don't think about it. And we have another question, which is, if the gold standard is not as reliable as clinical practice, how would you compare machine learning algorithm validation experiments against a not so great gold standard to the FDA? Read that again. Well, I mean, the FDA has been dealing with this problem for a long time, right? I mean, every time there's something new, there's some not so great gold standard. Let's say you did something that was really novel, you were going to, be able to tell if if there was a new disease, and you were going to be able to have a genetic test for it. And it's a new disease. There's no gold standard. I mean, this has been going on for forever, and you do the best you can. You'll use the best gold standard that you can come up with. It might be clinician's judgment. It might be all the various gold standards together. I mean, one of the big things you do is you use multiple gold standards, multiple pathologists. It might be pathology plus a lab test. It might be a lab test plus statistical arguments plus just professional opinions. So this is an old problem for the FDA, and you can't do what you can't do, but be creative in terms of trying to make the gold standard as good as you can. And the FDA knows that as you learn more, they'll accommodate that. All right, next slide. Right. So is metadata available, reliable, or helpful? So metadata is data basically based on other things. And of course, the beautiful thing about machine learning is that you can put in all kinds of information. As a matter of fact, there are success stories about things like trying to predict elections, right? And they put in, they have billions of records. Google has billions of records, and they look for like terms that people are looking up, and they look for things that you wouldn't necessarily think of as in a diagnostic, and it turns out that reflects some kind of an opinion of the general population. So metadata is the same thing is true with medical devices. You can an example of this might be you have a disease state. Let's say it's this is actually one that I worked on. It was trying to do ECG data, and age mattered. Or there's another one that it worked on that was trying to determine whether your heart was showing damage from pacemaker signals. And things like age and body mass index mattered. It turned out that when you gave it the information, that kind of data was helpful. So in general, you throw in all the things you can to see which ones are helpful, with the proviso that you're not giving it too much. There are times when people will create features or feature spaces, and they'll try 10,000 features. Well, if people underestimate the randomness of probability, and some of those are just going to randomly align. So that's a problem when throwing too much data at a problem, because then you'll overtrain to it. You'll find exactly the one that randomly correlated. Like, well, what's the famous one? Kennedy had a secretary named Lincoln, and Lincoln had a secretary named Kennedy, or something like that. And that's just it's just a random event. But if you look at enough things, you're going to find correlations, but they don't hold up in the long run. Okay, so let's keep going. So how do you know you're done? You're done when you add more data and the classifier doesn't change. So you do the training, you do it again, and the classifier doesn't change. And that includes you'll add more data, you'll retrain, you get a new classifier, the classifier doesn't change, and the result doesn't change. That's when you know you're done. I've never really seen that happen much, because usually we haven't had enough data to converge 100%, but it's close enough that it doesn't matter. Next slide. So what are some techniques? Don't overtrain, don't give it too many variables, don't give it too dynamic a range. In neural nets, that means don't make your neural nets too deep, and too many of them, and because it'll find results that aren't true. Next slide. Use repeatable instruments when you can, and try to train your operators so that the variability is minimized, so that do a good job at defining your training mechanisms. Next slide. Repeatable instrumentation. I just wanted to make an example of what happens. This is an example as you had blue and red, positive and negative, and then you get a new batch of instruments and go to the next slide, and all of a sudden your results are different. That's what happens when you don't have repeatability. And so just to reiterate, in a machine learning application, if you give it enough data, it will accommodate the new data. You can retrain it, but it's usually at a cost of reduced performance. So repeatability is often overlooked. People will use consumer devices. They're not controlled. You don't know what you're going to get. You don't know how they're going to change over time. It can be problematic. So look for repeatability when you think it might matter. Next slide. Pre-training with simulated data, another technique. We have an application where we're trying to use it's near infrared spectroscopy, something that you would use to measure oxygenation, tissue oxygenation levels. We've generated data sets from simulated data. The beautiful thing about optics is it simulates pretty well. So there's basic simulations, and then the trick is to make the simulations be as close as to reality as you can. That means noise, artifacts across the whole variety of morphology or disease states, and you start to get pretty complicated modeling, and then you can pre-train your classifier. What it's doing essentially is saying, I'm not going to have enough data to really train, so I'm going to fake some data and let it do some of the training on that, and then you fine tune it with the real data. And you can do that either with simulated data or simulated or similar data. And I used the example of cats versus dogs. So we have another question. It said, your learning data set includes certain medical data, but in real life, one or more of the input data is not available. Now what? Oh, that's a good point. It does happen, and there are times when it's a problem, but classifiers don't always die because they don't have a particular input. You can put it in sometimes in NA, and you might get a less accurate result, but it's still a result. So it depends how much that particular data matters. So, but sometimes it matters a lot, and you just have to get all the data in. It really depends on how important that metadata is. All right, next. So I'd be remiss if I didn't bring up, things like that machine learning has really changed a lot in the last couple of years. One of the things that you can do, in the past, much of the setting up of all the searching was done manually, and then it would search over this big space automatically. But nowadays, like for example with AWS Autopilot, it will not only search the space, it will search the way to search the space. It will change, the say for example the neural net architectures, or not. It's not, or even don't try neural net. It'll try tree structures, and it'll see which of these does the best job, and that just makes it a lot easier, compared to how much manual work used to be. And again, back to metadata. metadata. Is that helpful? So next slide. One of the tricks for sure is doing what you think is going to be useful. In other words, let's say, for example, you can have a completely open-ended machine learning algorithm. So that might be, for example, just giving data into a neural net, and it also might be giving it some predetermined features that doctors have been diagnosing with for many years. I mean, let's say that doctors have been looking at cancer slides for many years and say, or cancer images in vivo. And there's a thing called a dermatoscope, and they look at it and they say, I don't think that's cancer. I think we should do a biopsy. So there are things they've learned to look at over the years. They've correlated with the disease states, and you can create features that do that. An image processing feature. Let's say, for example, in another one, maybe a good one is melanoma. There's little circles in normal tissue that are called rete ridges, and when those break down, it means a certain level of disorder. So we created a rete ridge circle feature, and that helped the search engine rather than having to find it on its own. And that's one of the ways that I have found to be really powerful. If you're going to look for a small signal in noise, let's say you're looking for a heartbeat and it's mixed into the noise. Well, let's say you have a heartbeat from a pulse oximeter. Maybe you can what you can do is you can use that heartbeat, with some kind of correlation function. You can draw it out of the noise. So there's techniques that you can use to help the classifier focus in on things that you think are probably useful. And if it doesn't help to be useful, then I won't use it. But in general, it's been makes a big difference in my experience. Next. Actually, go back to one more thing, and that's pre-processing. Pre-processing can mean removing irrelevant information like noise, cycle noise, but it can also mean calibration. Calibration is a way to take out unwanted information and noise from your instruments. So it might, for example, let's say you have an instrument and it has a offset and gain problem, variability. You can calibrate that out, or something like that. There's lots and lots of different kinds of calibration, and you can. It's a whole art form to be able to calibrate your instrument to cause higher repeatability and to take out noise. So that's a really important factor. And then data normalization is something that's almost always done in machine learning. Next slide. Bob, you want to take this one?
Yeah, so on the, we just thought we'd be remiss if we didn't say something about what the FDA is looking for. So FDA has a document here, you all have a copy of this. Their proposed regulatory framework for modifications to AI ML based software as a medical device. So this is something that they published back in 2016. They were looking for input from people. So it's not completely settled, but usually the direction that they suggest is the direction that they want to go forward with. And it's based on standards that they have followed, historically. The examples they give you for these software as a medical device include diagnostic imaging software for cancer, or embedded software on a device that is computing the probability of a patient encountering a heart attack, or predictively having a heart attack. So any software like that would be included in their scope. So it's both devices and software only type applications. And two things that they talk about are this life cycle process, and they actually give some good examples of that on their guidance website, and that we'll have to have not only a life cycle process, but also a algorithm change protocol, as they call it, which is a risk-based thing. And if I scoot ahead to the next slide, there we go. They've actually provided that there's this international group that they've put together for software as a medical device, where, as usual, 1 is the lowest risk, 4 is the highest, and they want every submission to consider whether the device in a future iteration could become a higher level of concern to the patient and thus to the FDA, so that you would have to treat the documentation. In fact, the link here is to the FDA website that will describe all the current thinking on this, and it aligns very well with what they have done throughout history. It's a risk-based approach. If you have more risk, they want to see that you're taking more care.
The FDA also has an interesting pilot program, which is about being able to be certified to update your own software updates. They recognize, for example, for both artificial intelligence and security issues, that it's not really practical to always be resubmitting data to the FDA. It takes too long for them to respond, especially for security things. So what they've gone to is a process-oriented approach, where they say, if you have a solid quality system and a solid process, and they kind of approve it, then we trust that your results are going to do essentially what you said they were going to do. So it's a pilot program now. I've been really interested in it, and we'll see how that plays out. But that's something that in machine learning is an interesting thing, because as things get more automated and updated, it would make it easier to implement and deploy updated classifiers or algorithms.
Yeah, and they even said, I mean, to that's a good point, Adam. They said that if they have a document, of course, called deciding when to submit a 510K for a software change to an existing device. And if you've modified the algorithm architecture or the test data sets, then you can pretty much be assured that it is something you'd want to submit a subsequent 510K for. And they'd say you'd be looking at performance change in inputs, or change in intended use. Any of those things certainly would trigger an additional 510K submission to the FDA.
So there's a question about qualifying for the pilot program. It was a voluntary, when I looked into it, there was a voluntary program where they were looking for a certain number of participants and you would apply, and they would essentially decide if they would let you in. This has been going on now for at least a year, so I'm not actually sure what status they are about taking new applications, but it's not hard to find. So if you look at the FDA and search for this program, I'm sure you could dive into it. Another question, and it's good that we're into questions because we're getting near the end. Are there instances where pre-processing, such as smoothing, can inhibit feature discrimination? Yeah, definitely. There's times when you are taking information out. So is that a benefit or a hindrance? An example of that is just recently. This happened just last week. We were talking about using a certain kind of pre-processing for doing principal component analyses to try to see if we could generate some features to get rid of a noisy algorithm, and it was this was an EEG signal, and basically using support vector, principal component analysis to get rid of breathing artifacts, and the breathing artifacts are very low frequency. So if we did a low pass filter, high pass filter, I'm sorry, which we did, then we were cutting out the main frequencies of the breathing artifact, but you would get the harmonics. So we decided not to do that. Let the PCA have the full signal, and then it would be easier to separate out as a principal component because it had the main harmonics of the breathing. So absolutely, you have to be careful. And this is sort of a trial and error kind of approach anyway. So you try it both ways and you see which one works. That's a big part of search engines, is you try all these different things and you see which one works. There's in the chat, there's well, I don't know if everybody can. Yes, in the chat, there's a link to the FDA pre-certification program. Thank you to Todd. who did that. Are there any more questions? Because we do have a few more minutes, and we got a great presentation award here. So thank you, Catherine, for that. It's always good to hear after you blabber on for about an hour. Someone is working with continuous data sets of 2 biological parameters. What ML algorithms would you recommend that I use to analyze my data? 2 biological parameters, multiple samples per minute. That's continuous data. So I guess that's a signal data. I'm trying to understand the question. I think that's a signal data. You're talking about if they have a signal that's coming in. There's 2, let's say it's ECG and I don't know, respiration or blood pressure. And they're saying like, what algorithms would you recommend? I mean, first there are, I mean, there's, I think when you're saying algorithms, I mean, first of all, one thing you could do is you could try to use just a neural net. A word of warning about neural nets is that you know you never really know what they're doing, and you can't push the sensitivity to 100%. By definition, you can't do that. As far as this one I did it, we couldn't do it. So if you want to train for 100% sensitivity, that's hard to do that with neural nets. But if you're looking for signal processing, and you wanted to find patterns in the whatever parameter you've got, you can use neural net, and it might find those patterns. Just be aware that it might not. I mean, it's a real common mistake that people say, well, I don't know, like, I mean, EEG, we're going to take EEGs and we're going to see if I hope there's let's say that there's either a disease state or we worked with some people who are working on toxicology, where if people were impaired or drug impaired, you don't really know a priori. You kind of have a hunch that you might even have a mechanism that there should be something there, or a spectroscopic signature from a molecule, but you don't really know if it's in there or not. So sometimes, just be aware that expecting it to do the hard part of finding the pattern, it may work and it may not. You just don't know. I mean, if you know that the signal is in there, then you have a good shot of picking it out. Assuming that there's not confounders and noise and all that stuff. But just one of my words of warning is, don't expect it to be so much smarter than us that you can't see it and you're not sure it's there. And you never know. You might get lucky, but you might not. So I think for just continuous signal processing, a neural net's not a terrible one. But I would say try to find features that bring it out. We have 1 minute left. Anybody?
You have several thanks there. So Adam, thank you for doing such a great job on the presentation.
Oh, so there's a question here about software as a medical device, and are these algorithms held to a different standard in regard to risks and performance metrics? I mean, software as a medical device is a funny thing, because many people use it. I mean, it depends. It's just like what Bob said. It's risk based. So if you're doing a consumer educational device where you're telling someone that that maybe it's time to look at their insulin, then and you consider the risk low, then they're pretty loose about it. But if you're using it to inform high risk procedures, or then it's really just risk based about software as a medical device. So
yeah, in this link on the on slide where is it, slide 23, artificial intelligence, machine learning, and software as a medical device. That's a link to you could just Google that. That's an FDA website. And it goes into all the details of how they're looking at this. So we'd be happy to, answer questions and provide guidance if you have follow-on questions afterwards.
Yeah, and this question was making a distinction between software in a medical device and software as a medical device. I think that much of the software as a medical device is kind of a newer thing, and they're finding their way. That's been my observation. And much of it is based on, like I said, sort of consumer-based things or less risky things. And if it's software in a medical device, I think it obviously is, affecting that device. So I really think it's just a function of the risk. I think the answer is still the same. I just wanted to, but the question was about that.
Well, we are at time. Thank you for this wonderful conversation. Thank you, Adam. Thank you, Bob. Oh, great. You put your contact information up there. Please feel free to reach out to them at Sunrise Labs if you have any additional questions. We will be sending out a recording of this conversation today to everyone who registered, and you will get a copy of the slides as well. So again, thank you everyone for attending. Thank you to our sponsor, Sunrise Labs, and we will see you again here next time. Thanks. Have a great day.